Okay, let's get started. Thanks everyone for your patience. And welcome to join us today. And thank you very much for attending Microfluidics webinar titled Microfluidizer High Shear Processors for the Vaccine Industry. My name is Yang Su and I'm the Manager of New Technology and Applications of Microfluidics. I will serve as your moderator for today's webinar. As you all know, many companies are currently working on developing vaccines in response to the global coronavirus pandemic. So I hope to use today's webinar to provide an overview of the different types of vaccines and demonstrate how microfluidizer technology can help manufacturing the main components of vaccines, which are antigens, adjuvants, and delivery systems on production scale. Before we get started, I'd like to go through a few housekeeping rules. We'll record the whole webinar session, so everyone will be muted during the entire webinar, and we appreciate that you continue to keep your lines unmuted. There will be a live Q&A session after the presentation. So if you have any questions, please submit your questions via the chat window. We will try to answer as many questions as possible. If we don't have time to go through all of them, we'll certainly follow up with you shortly after the webinar. Again, we have already started recording today's webinar, and the recording will be hosted on our blog. Now, I'd like to introduce you to today's speaker, Steve Masidi. Steve is Microfluidics Director of Machines. He has 15 years of experience across a wide variety of applications through closely working with customers in numerous proof of concept test process development, optimizations, and consultants. Now, without further ado, I'm going to hand it over to Steve.
Thank you very much, Young, and thank you to everyone for taking the time to join this webinar and hear about our technology and our applications. This is the agenda. I'm going to start talking about who we are as a company, and then I'm going to dive right into what are the main components of a vaccine, and get to talk about how vaccine technology has evolved over the years. Then I'm going to discuss what is unique about our microfluidizer technology, and then we have 3 application examples, as Young mentioned: vaccine adjuvant nanoemulsions, lipid nanoparticles, and cell disruption for production of proteins. Then I'm going to talk briefly about our product line, and we should have plenty of time for questions and answers at the end. So this is just a little bit about microfluidics. We have been around for about 40 years, making microfluidizer processors, which are based on our interaction chamber technology. We're located just outside of Boston, but we have global sales and support in 47 countries with 4,000 processors sold to 2,000 customers globally. We're part of IDEX Corporation. We're in a group called the Material Processing Technologies Group, along with 3 other companies: Quadro Engineering, the Fitzpatrick Corporation, and MadCon. Our equipment is primarily used for R&D and manufacturing of pharmaceutical ingredients and also vaccines. And our core competence is in applications and machine design. And what I really want to distinguish is that we are not vaccine experts. We're not epidemiologists. We don't have any kind of political leanings or any opinion about which vaccines are going to win the race to the market. I actually, think that it's this pandemic is, one of the silver linings is that a lot of vaccine development and research is going to come out of this, and I'm very confident that at the end of the pandemic, we're going to learn a lot. We're going to know a lot more about vaccines than we do now. So that's it. We're not here to make any political statements or advocate for one type of vaccine over another. So this is very high level, but just to make sure that everyone's on the same page, what is a vaccine? A vaccine is a drug that forces a patient's immune system to develop antibodies to a specific pathogen. So this is a picture of the virus, and On the outside of these viruses, there are these spikes. It is very common to make spikes the target for antibodies. So if your body has antibodies that will bind to these spikes, the virus will be inactivated. So how do we force our immune system to create these antibodies? The first thing you need is some sort of antigen. An antigen is an entity that will cause the production of antibodies to bind to that specific entity. I'm going to talk a little bit more about different types of antigens in a minute. Another important aspect of vaccines are adjuvants. Adjuvants are immune system stimulators. They don't cause any diseases or any antibody production, but they just cause the immune system to activate and will cause more recognition of antigens. And then the other part that I'm going to talk about are delivery systems. Especially the most recent amino acid type vaccine antigens require delivery systems in order to deliver the active to where it needs to go and potentially protect it so that it is stable for long enough to be active. There are two main types of delivery systems. Lipid nanoparticles are the ones that we're going to focus on, but we've also seen a lot of activity in AAVs, adeno-associated viruses. So these are the different types of antigens that the main types. We're going to kind of break it into three main types. The first type is the native virus type. So basically what you do is you take the virus. This is another image of a virus. This is the actual SARS-CoV-2 virus that has been imaged by the University of Washington. And what companies will do is take this virus and either inactivate it, so they'll kill the virus so that it doesn't replicate, or they'll attenuate it so that it is much less active and reproduces at a much slower rate. One example of this in effect is Sinopharm out of China has a inactivated SARS-CoV-2 virus that is currently in clinical testing. These are typically slow to develop. It takes a while to isolate the virus and kill it and make sure that it is it won't replicate, but also will act as an antigen. The good thing about this type of vaccine is these are currently produced on a very high scale. They're typically produced in chicken eggs, and there's a lot of infrastructure to produce these types of antigens on very large scale. However, the production speed is very low. It takes a while to grow these inside of chicken eggs, and then downstream processing in order to concentrate and isolate the antigen takes a while. But there is quite a lot of these types of vaccines currently on the market that have been FDA approved. The key application that Microfluidics sees for this type of vaccine are adjuvants. Some of these vaccines require adjuvants, some do not, but that's one that we typically see. The next type of vaccine is recombinant protein-based. So rather than administering the entire virus, you can just take the specific part of the virus that you want the antibody to bind to and produce that. So this is the spike protein that you probably heard about, the S protein. And this has been isolated and can be manufactured in bulk through recombinant processes. It's grown in a couple of different cell lines, and there's a very large partnership that's been in the news that's developed between GSK and Sanofi. So Sanofi is producing the protein, and GSK is producing the adjuvant. It's one of the largest pharmaceutical collaborations of all time. The development speed of these types of antigens is slow. It takes a while to create this protein, make sure that it's accurate. It's actually done using microscopy and then recombinant processes. There is very high scale for this type of antigen already on the market. A lot of these have been produced in the past. The production speed is extremely fast. It's about 10 times quicker than for the native virus type. And there have been a lot of these types of vaccines that have been approved on the market as well. And the key applications that we're going to talk about are both antigens and adjuvants. The third type of vaccine is nucleic acid based. So for this type of vaccine, the either DNA or RNA is delivered to the body, and then this RNA will code the protein that is used as the antigen. So rather than delivering any sort of protein, you can just deliver this amino acid and your body will naturally produce the protein. Moderna was very famous in the news very early on in early March because they brought a vaccine to the clinic within 2 months of isolating the protein, which is extremely fast, much faster than any other type of vaccine that can be produced. The current scale for these types of vaccines is low. Typically only small batches are produced, and the number of doses that can be produced is quite low based on current technology and availability of raw materials. But the production speed of these is very fast. You can produce these, in a matter of hours rather than, days or weeks for the other types of vaccines. Another hurdle that will have to be overcome during this process is that no vaccine using nucleic acid-based technologies has ever been approved for pandemics. So if one is approved during this pandemic, it will be the first. But that's always a big hurdle that needs to be overcome. Currently, according to the World Health Organization, there are about 120 different vaccines that are currently either in clinical trials or preclinical, and they range across all three of these different types in about equal proportion. So it's about a third native virus, a third recombinant protein, and a third nucleic acid. So all three of these types of vaccines will likely be used to combat this pandemic. So real quick, what makes microfluidizer technology unique in the market? So microfluidizer processors ensure that every milliliter of material gets the same high shear treatment, regardless if you're processing a very small 1 milliliter batch or if you're scaling up to thousands of liters. And what that means is our technology is used to produce nanoemulsions that can be sterile filtered quite easily. We can perform cell disruption at the lowest energy levels so that you can disrupt the highest number of cells with the lowest amount of protein denaturing. And we're also used for uniform particle size reduction. That doesn't really apply to vaccines, but it is a common application of our equipment. So let's hop into the case studies. The first one that we're going to be talking about is vaccine adjuvants. So as I mentioned earlier, adjuvants are ingredients added to vaccines to induce a greater immune response. This graph shows the number of antibodies found in the blood as a function of time for 4 different types of vaccines. So there are two vaccines with no adjuvant, this one vaccine with the triangles. This has an aluminum-based adjuvant, and this vaccine up here has the MF59, which is a nanoemulsion-based adjuvant. There were 3 administrations of this vaccine, and this vaccine was to produce immunity to hepatitis B. So what you can see right away is that after the first administration, after the second week, there was already about 50 times more antibodies produced by the vaccine with the nanoemulsion-based vaccine adjuvant. And that continued throughout the period of analysis. So after the second administration, it was only about 100 times more than the other types of vaccines. And after the third administration, you can see that it was about 10,000 times more antibodies generated or available in the blood with the nanoemulsion adjuvanted vaccine. And that means a couple of things. Number one, it is more antibodies means that your body will be more likely to fight off infection. But number two, if you only require, 10,000 units of antibody per milliliter of blood, you can get away with fewer administrations of the vaccine, which are really important. We're talking about vaccinating a population of, 7 billion people. If you have only 1 or 2 administrations versus 3 or 4 administrations, that really reduces the effort. Fewer administrations is much more desired. Additionally, if you're relying on the antigen to produce the immune system response, you probably require a higher level of antigen in the vaccine to generate the same amount of antibodies. Using a vaccine adjuvant, typically you administer a lower dose of the antigen and get the same therapeutic effect. So we're talking about the cost and bandwidth for producing these antigens. If you use an adjuvant, the amount of antigen that is produced can result in a larger number of doses. So these are the 2 main nanoemulsion-based vaccine adjuvants on the market. MF59 is used in Fluad, which is a seasonal flu vaccine by Seqirus, and GSK produces this AS03 for a few different types of vaccines, including pandemic vaccines. So this is how this case study is about production of a vaccine adjuvant nanoemulsion. This case study was done in collaboration with the Infectious Disease Research Institute out of Seattle, Washington. And if you're interested in learning more about this case study, I urge you to check out the webinar that we conducted with Chris Fox from IDRI called Vaccines for Global Health. So the goal of this case study was to reduce the size of the nanoemulsion to a D90 of less than 200 nanometers. This was done by introducing a crude nanoemulsion to the inlet reservoir of a microfluidizer processor, which has a constant pressure intensifier pump, which is important to achieve uniform processing. And then it is forced through our fixed geometry interaction chamber. For nanoemulsions, we use this Y-type interaction chamber. The material is forced in here to the inlet, and then through these microchannels. These microchannels are typically 100 microns or so in nominal dimension, which is about the size of your hair. And when you force liquid streams through these microchannels at very high pressures, up to 30,000 psi, which was used in this case, you get extremely high velocities. We're talking 400, 500 meters per second. Those velocities then in turn create 3 different types of forces. The first is shear, the second is impact, and the third is energy dissipation. And those 3 forces are what we use to create these very fine nanoemulsions, which are of the desired size, less than D90 of less than 200 nanometers. So why is a D90 of less than 200 nanometers important? It's because of sterile filtration. These vaccines need to be administered typically via intramuscular injection. And in order to do that, you need to have a sterile product, and sterile filtration is by far the easiest and least expensive way to sterilize these types of emulsions. So in order to evaluate the particle size requirement for nanoemulsions, we have a collaboration in place with Paul Life Sciences. They make sterile filters. And if you're interested in learning more about sterile filtration and the impact of particle size, there's another webinar on our YouTube channel called Production and Sterilization of Biopharmaceutical Nanoemulsions Using Filtration. So I urge you to check that out if you're interested in learning more. But the crux of the study was we produced nanoemulsions with various particle sizes and filtered them through a few different sterile filters. The most important sterile filter that we use is this EKV filter. It was found to be the most effective for sterilizing the emulsion and removing all of the bacteria. And you can see that with a D90 of about 240 nanometers, you get a very low throughput through this filter. If you reduce the size by about 15%, you get about 10 times more throughput through the filter. And then if you reduce it another 10%, you get another 2 times more throughput. So if you get your D90 down to around 200 nanometers, we believe that you will be able to successfully filter your emulsion. So the work that we did with IDRI, we processed the emulsion on our 110EH microfluidizer processor, which has a flow rate of about 300 mils per minute. And after process optimization, we found that you really needed about 2 passes at 30,000 psi in order to achieve the required particle size distribution. And then a third pass was also performed, but you can see the difference in particle size between the second pass and the third pass isn't all that much. So the next thing we needed to do was scale this up. So one of the key benefits of the microfluidizer processor is the inherent scalability of the technology. We're able to take these microchannels, align them in parallel, and then put them in a single multi-slotted interaction chamber. And then we use a larger pump, and we can pump at a higher throughput through these microchannels. And since the microchannels have the exact same dimensions and you process at the same pressure, you get the exact same shear force, the exact same impact force, and the exact same energy dissipation, which means that you get the same particle size. So we ran production scale tests on the 7250, which is a 50 horsepower microfluidizer, which runs at 5 liters per minute at 30,000 psi. And these were the results that we achieved on the lab scale. And I'm going to overlay the production scale results. So you see the first pass on the production scale is a little bit smaller. The second pass was almost identical, and the third pass was virtually identical to the lab scale. So it was a successful scale-up experiment. And yeah, if you're interested in learning more, please check out the IDRI webinar. All right, the next application I'm going to be talking about is lipid nanoparticles. Lipid nanoparticles are vesicles that contain a lipid-based surfactant, most commonly a phospholipid. This is an example of a phospholipid. This is DSPC, which stands for dipalmitol phosphatidylcholine. It has this phosphorus group here that's common among phospholipids. And then these lipids will assemble to form nanoparticles, and these lipid nanoparticles are used for antigen delivery because they increase the circulation time in the body and help deliver actives to the target site. There are many different factors to consider when designing a lipid nanoparticle system. There are many different types of phospholipids. I'll talk about this a little bit more on the next slide. Carrier oils are often important to maintain the rigidity of these lipid nanoparticles. They can either be solids or liquids at room temperature, and that has different effects. There are many different types of surface modifiers that can be used. Most commonly, PEG is used to shield the lipid nanoparticles from take-up from the MPS system. The antigen type is also important. They can be RNA-based or DNA-based or protein-based. And there are different processing methods that can be used. The microfluidizer technology is one type of processing method, but there are others as well. So this is an example of a lipid nanoparticle. This type of lipid nanoparticle was produced by IDRI, and if you want to read more about it, it was published in this in the molecular therapy. But it has this DOTAP phospholipid, which is a cationic phospholipid, which means that the RNA, which is negatively charged, will adhere to the surface of this phospholipid. DOTAP stands for dioleol trimethyl ammonium propane, and it is a common phospholipid for RNA delivery. It was also used two different types of surfactants. SPAN and tween are both polymeric surfactants. They used squalene as the carrier oil, which is the same oil found in MF59, and that means that this particle will not only be a carrier for the antigen, but also will have an adjuvant property. So it's not only is it a lipid nanoparticle to carry the antigen, but it will also increase the immune response. They also used Dynasan 114, which is a solid lipid at room temperature, meaning that this lipid nanoparticle will be more rigid. And then the blue is obviously the buffer. So this nanolipid carrier system, which they call it, will be produced in one vial, and then this can be mass produced and stored for pandemics, and then you can have the RNA delivered in a separate vial. You mix them together and administer the vaccine. So there are many different types of outputs that these lipid nanoparticles can have. They'll have different activity depending on these inputs, different immunogenicity, if you're trying to use them as an adjuvant or not. There are many factors that will influence the circulation time in the body, or whether or not there will be a depot effect. And also, the processing method will determine the particle size. So this is the most common way for production of lipid nanoparticles. It's a 5-step process where you make a solid mixture of the phospholipid, the carrier oil, and any actives that you want in a solvent. You evaporate the solvent, add buffer, you warm and vortex to hydrate, and then you can add your antigen, and you get these nice big multi-layer vesicles. They're typically in the range of 1 to 5 microns. You pass them through a microfluidizer processor, and in the very similar method to producing the nano emulsions, and you get these nice small unilamellar vesicles, which are around 100 nanometers in diameter. So this is a case study which we ran in collaboration with the University of Strathclyde, and we were able to produce a webinar called Formulating Liposomes and Importance of Vesicle Size Control in collaboration with Ivo and Perry. And we ran 4 different formulations of lipid nanoparticles, and we process them to achieve uniform particle size, all in the range of about 100 nanometers with polydispersity right around 0.2, so they're all very small, very uniform in size. The only difference is the formulation. So this is just DSPC and cholesterol, which is a neutral charged phospholipid. And then we ran two different anionic phospholipids. PS is just phosphatidylserine, and DMPG is dipalmitol phosphatidylcholine. The difference between these two phospholipids is phosphatidylserine has a high glass transition temperature, and DMPG has a low glass transition temperature. So these will have different rigidities in the system and can change the delivery effect. And then we also ran formulation with DOTAP. And this is, again, as I said, a cationic or positively charged phospholipid. And what we found is that the formulation had a really high impact on the ability for the phospholipid to leave the active site. So this is a graph that shows how the phospholipid particles, the lipid nanoparticles, migrated away from the injection site after about 200 hours. So you can see the negatively charged phospholipids are these two, the red and the green, and those had the quickest transfer from the active site, and had the most complete transfer from the active site throughout the study. And then the cationic took a lot longer. So this is what we would call a depot effect, where the lipid nanoparticles remain at the injection site and then slowly elute to the target site. And different vaccines will benefit from very quick elution from the active site, and some will work better if there's a depot effect. A depot effect can be used as a time release effect, but in order to have the benefit of this time release effect, the immunogenicity needs to be high enough of these cationic particles. So there needs to be enough of a release in the system in order to cause the desired effect. So again, this is these types of delivery systems are quite complicated and require a lot of R&D in order to achieve the desired effect. So the third case study that I'm going to talk about are recombinant protein production, and the only part that the microfluidizer technology plays in this process is cell disruption. Most recombinant antigens are produced in either E. coli, yeast, insect, or mammalian cells, and most recombinant processes require cell disruption to extract the proteins from the cells. So this is a case study of bacteria cells. This is E. coli. It's processed in very similar methods to emulsions. The main difference is we use typically a Z-type interaction chamber rather than a Y-type. So the material is forced through a single microchannel and forced to impinge upon a wall. So the impact force isn't between the two streams, it hits a wall, so the cells are broken in that manner. Another aspect of the technology that I want to highlight here is the heat exchanger. All of our processes do impart energy into the material, which will cause an increase in temperature, and that can be bad for temperature sensitive proteins. So it's very important that a heat exchanger is used immediately downstream of the interaction chamber. And here you can see this is kind of what the bacteria sample looks like after just one pass. You can see virtually all of the bacterial cells have been broken up. So different types of cell lines require different shear rates in order to break them. You can see that bacterial cells require high levels of shear, 500,000 or 600,000 inverse seconds of shear, whereas yeast cells are much tougher to process. They require, often high levels of shear and occasionally multiple passes in order to achieve very high levels of cell disruption. Using our fixed geometry microchannels, it's very easy to calculate shear rates. Shear rates are calculated based on a velocity gradient across a distance. So shear rate is the maximum velocity, which is in the middle, minus the minimum velocity, which is at the wall. We assume it's 0. And you divide that by the distance, and we get a wide range of shear rates. We can control shear rates using two main factors. The first is the pressure. So we can control the shear rate by the pressure. The second is the geometry of the microchannel. So what is the size of the microchannel? So for bacterial cells, we typically use this H10Z at around 20,000 psi to get the desired shear rate. For mammalian cells, it's much lower. We generally use this H30Z or this L30Z at around 5,000 psi to get the desired shear rate. So I have a few examples of different cell lines. This is S. pombe, which is a very hard to disrupt cell line. And we used, a very high shear rate through our G10Z interaction chamber at 30,000 psi. And you can see that for each pass, we ruptured around 60% of the cells. And after 5 passes, we were able to get over 95% lysis. And for this type of process, we typically recommend between 3 and 5 passes. Whereas for mammalian cells, they're much larger. And, they're typically in the range of 10 to 20 microns, and you can get virtually 100% of cell disruption within 1 pass at relatively low shear rates. So that's it for the application examples. I'm going to talk really briefly about our product line. We have a very wide range of microfluidizer processors depending on what you're trying to do. This is the LV1, which can process samples as small as around 1 or 2 milliliters. So it's very good for R&D. And then, we have benchtop units, which are good for processing batches of around 50 to 100 mils. These are the pilot scale, which can process, in the range of ½ liter to 1 liter per minute. And then we have a production line, which can process 5 to 15 liters per minute. So they can be used to produce very large batches of thousands of liters. We are also very familiar with the CGMP requirements for, the registration bodies, including the FDA and the European bodies as well. And we have processors that are designed to meet those requirements. So for starting at the 110P, which is a benchtop unit, all the way up to production, we offer 3 different types of CGMP compatible pieces of equipment. The basic ones just are drainable. They have material certs and electropolish and passivation. Our enhanced versions have 21 CFR Part 11 data acquisition, which is becoming more and more important as considered by the regulatory bodies. And we can also produce these pieces of equipment with steam in place, and they can be used for aseptic processes. So if you have a process that you cannot filter, terminally filter, you can run the entire process in an aseptic environment, which is obviously a lot more challenging and more expensive, but it is certainly an option with our technology. All right. So that wraps up this portion of the presentation, and I'll turn it back over to Young to host the question and answer portion.
Thanks, Steve, for a very informative presentation. If you missed before, please use the chat window to submit your questions. We're open for questions now. Um, we did receive several questions prior to the webinar. So while people are typing their questions, let me bring those up. So the first question is, what's the impact of particle size on the efficacy of lipid nanoparticles?
Good question. So it can have it can affect the ability of the lipid nanoparticle to transfer away from the injection site. Typically, I believe this was one of the parameters that we studied with the University of Strathclyde, and they compared two different types of lipid nanoparticles. One was small, around 100 nanometers in size. The other was much larger. It was around a micron in size, and the 100 nanometer lipid nanoparticles did elute away from the injection site as presented on the slide earlier. And the large lipid nanoparticles, they formed a depot and did not elute very effectively at all. I believe that after the 200 hours, they remained pretty much exclusively at the injection site.
Thanks, Steve. The next question is Microfluidics focused only on vaccines to fight COVID-19, or are there any other drugs for treatments of COVID-19?
Oh, yeah, that's a good question. Microfluidizer technology is used to produce other types of drugs too. The most important one right now that we see is propofol. Propofol is an anesthetic, and it is used to help patients as they are put into induced comas to go on respirators. So propofol is something that is another nanoemulsion technology. So it's a very good application for our equipment, and we're seeing there's a global need for propofol currently. So we're seeing a lot of companies trying to make propofol at a greater scale. There's other small molecules that are of interest that have just been developed. That haven't been clinically approved yet, that we're seeing trying to get through the approval process in an expedited form. One of them that we saw is to help prevent the cytokine storm, which is presenting in some COVID-19 patients. So yeah, it's not just vaccines that we're focused on, but also actives, and then also recombinant technology for protein production isn't just used for vaccine adjuvants, but it is also can be used for the testing for COVID-19. So for running these tests, some of the reagents that are produced using recombinant technology. So we are seeing a lot of customers trying to produce these at larger scales too.
Thanks again. There's another question. The route of delivery for vaccines such as intravenous, sub-Q, or intramuscular seems crucial. Sometimes mimicking infection route or infections helps. Any guess for the delivery route for the COVID-19 vaccines?
It's a good question. I think probably different vaccines will use all of those routes. I was going through the World Health Organization's vaccine list, and some of them claim the delivery system as a critical part of the vaccine. So I know that the vaccine that's being produced by GSK and Sanofi will be intramuscular, but I also know that there are new administration technologies that are being developed too, including microneedle arrays for patches and electroporation. There will probably be some that will be administered via IV, but that's more rare because it's a lot harder to administer via IV kind of on a mass scale. It requires a lot more skill than the intramuscular or the patch methods. But I think that there of the hundreds some odd, they will all probably use different administration routes.
All right, the next question is around, I think you touched on that a little bit, around cooling efficiency for products. So how efficient is the heat exchanger, for example, with the LM20 microfluidizer processor? If you pass 3 to 5 passes at the max pressure of 30K to break yeast cells to achieve over 90% of rupture, that's going to generate a lot of heat. Is that going to denature the protein, even with the cooling ice between? Because if the sample volume is big, it can take quite some time to process all of the samples. Is there a 40K instrument for research purpose, for maybe help to reduce the passes to 2 or 3?
Good question. There's kind of a lot to unpack there. So the LM20 is it's the benchtop model. It's this one right here, and you can see that it's there's a cooling coil and an ice tray, and yeah, I think that's a perfectly valid point. If you're processing a large sample like a liter for 5 passes, 5 liters, it will probably be very important to kind of keep this tray packed with ice water in order to prevent the sample from heating up. This is probably not the best piece of equipment for that type of process. It would be better to use, the 110P with an integrated heat exchanger, or the 110EH, which also has an integrated heat exchanger, in order to be more sure to control the temperature continuously throughout the process. These ice baths are nice because they have very small holdup volumes, which is good for R&D, and you don't require any kind of recirculation system. But for larger batches, the heat exchangers are a lot more effective. But yeah, for the LM20, if you keep the cooling bath filled with ice, it should be able to maintain the low temperatures required in order to preserve the temperature-sensitive proteins, even with very high batches. The question about 40,000 psi is it's a good question. We have sold equipment that goes up to 40,000 psi in the past, all the way up to the mid 2000s. I think we had a machine called the 140K, which could produce 40,000 psi, and I think we sold that until about 2006. And what we found is that processing at pressures above 30,000 psi had 2 main issues. One is the temperature was a lot higher, and although it might reduce the number of passes, the higher temperature would cause higher levels of protein denaturing. And the other bigger issue that we saw with 40,000 psi was related to the robustness of the process piping. All of our process piping is rated for 60,000 psi, and our equipment has a maximum pressure of 30,000 psi. But there is a cyclic load, especially of our lab scale equipment. And even at 30,000 psi with this cyclic load, the 60,000 psi process piping will eventually break. And at 40,000 psi, it breaks an order of magnitude more quickly. So that's the main reason that we stopped producing equipment that goes up to 40,000 psi. The temperature is one. We didn't see that much benefit above 30,000 psi, but it's really the robustness of the equipment at 40,000 psi that made us decide to discontinue selling equipment at that pressure.
Thanks, Steve. There's also a comment provided by Bill in the chat window. He was saying he has experienced that for each cells, if you don't freeze them before rupture, you may be able to achieve a good rupture at lower pressure. So that's maybe a helpful hint. So the next question is, with extraction of membrane protein, do multiple passes help reduce the membrane fraction size? For example, create smaller, more homogeneous membrane fraction size, which may help facilitate subsequent membrane protein extraction?
It's a good question, and the answer is yeah. Typically for membrane proteins, we would recommend processing at even higher shear rates. So, for bacterial cells, we typically recommend 30,000 psi. If you want to break up the membrane, we'd recommend processing at the higher shear rates of like 30,000 psi. And but at that pressure, typically 1 to 2 passes does break up the membrane to relatively small size and allows for further downstream purification from our experience.
All right, the next question is the most infectious agent seems filterable. Scrappy type misformed proteins may be generated and make it through. Can this be monitored?
Sorry, you kind of broke up there in the middle. Could you repeat that, please?
Yeah, sorry. Let me repeat the question. So
maybe I can pull it up on the chat window.
The question asks, most infectious agents seems filterable. Scrappy type misformed proteins may be generated and make it through the filtration step. So can Rose misformed protein be monitored?
I don't know. So yeah, it's a good question. I think yeah, I think we'll have to look into that and get back to you. I don't have a good answer to that question.
Yeah, I don't either. I think this is a bit outside of our processes, and yeah, we'll look into that and get back to you.
Yeah.
All right. The next question is, what are the pros and cons for the microfluidizer processor? For example, the LM20, if you compare that to constant system for research scale work.
It's a good question. I don't have a whole lot of experience working with constant systems. I know that their technology is also has a constant pressure pump. I believe the main difference is that constant systems has a single type of microchannel. It's not really a microchannel. I think it's like a circular hole, and I believe it's significantly larger than the microchannels that we use. So I believe that the main difference is that with the microfluidizer technology, we're able to achieve higher shear rates at similar pressures. So since in order to rupture E. coli, you need 5 to 600,000 inverse seconds of shear, which we can achieve at 20,000 psi using a 100 micron chamber. I believe the constant systems orifice is about double the size. So you would need a higher pressure to achieve the same amount of disruption. And at that higher pressure, you're going to get a higher temperature. So to be honest, I don't have apples to apples comparison between the technologies, but I believe that the microfluidizer technology has a more flexibility in terms of the size of the microchannel that can be offered.
Thanks again. I have another question pre-submitted, actually kind of talk about a different method. If produce lipid nanoparticles using LabonAChip technology, Is it possible to transfer the technology to the microfluidizer technology? And if yes, how?
Oof, that's a good question. So yeah, so LabonAChip Technology is it uses a different method in order to produce very small lipid nanoparticles. Instead of taking these multilamellar vesicles and reducing them in size, they use anti-solvent precipitation in order to produce inside these very small microchannels in order to produce the lipid nanoparticles. And we have in the past taken the same ingredients that are used to produce lipid nanoparticles using LabonAChip technology and produce them using the microfluidizer technology, but the process will be different. LabonAChip technology requires kind of very low concentrations of lipid nanoparticles. The microfluidizer technology, usually we work with higher concentrations, not always. With the microfluidizer technology, it's a lot more flexible, but the hydration method will need to be different. Depending on your ingredients, you might need to use the thin film method or hydrate them in a different way. But we have done this in the past successfully, and we believe that most lipid nanoparticles that can be produced using LabonAChip technology can be produced using microfluidizer with a little bit of process development. And that's really important because LabonAChip technology isn't very scalable. It would be very difficult to achieve large batches with LabonAChip technology. So we believe it's a good application for our equipment if you want to scale up a LabonAChip process. We recommend that you reach out to us and we can set you up with a proof of concept test in our lab to see how it works.
Great. Kind of a related question to that. Would the high pressure and high shear forces introduced damage the the nucleic acid, DNA, RNAs, or the proteins that needs to be delivered?
Sure, good question. The answer is it can. It depends on the molecular weight of the molecules. We do a lot of work with molecular weight reduction of polysaccharides. That's another application for our equipment. And if the molecular weight of the molecule is, in the range of hundreds of thousands of kilodaltons, it can change the properties. But for most proteins, they're a lot smaller than that. Nucleic acids can be in that range, but the plasmid type nucleic acids that are typically processed are not so are not as affected. So it's not as impacted, but it can. So if we do see that the nucleic acid is being impacted by the shear, we try to achieve the results with a lower shear rate by lowering the pressure or by increasing the size of the microchannel.
Thank you, Steve. I think we're at the end of the hour now. With that, I'd like to conclude our webinar today. I'd like to thank everyone again for joining the webinar, and thanks, Steve, for giving such an informative presentation.